In the following video, we'll be summarizing the scale-invariant movement encoding in the human motor system.
This study asks, how does the human motor system encode our incredibly diverse motor repertoire in an efficient manner? What previous research has shown is that our handwriting remains geometrically the same regardless of the size and scale of geometric shape. This feature is to be taken as evidence of hierarchical organization of the human motor system. This enables efficient encoding for the motor system since movements are planned and then translated to precise kinematics. Earlier research led by Schneider has suggested that the anterior intraparietal sulcus, aka the AIPS, and adjacent parietal regions are high-level processing areas. They contain neural populations and plan future movements and encode movements according to abstract intentions and goals. In comparison, the M1 region was only thought of as a low-level area. The contemporary model had it only encoding instantaneous dynamic and possible kinetic movement variables. Further research led by Hatsopoulos into the M1 area had apes perform elaborate movements. The study's results suggest that the M1 neurons encode more complex time-evolving trajectories or goal-directed actions. The recent research suggests that the hierarchical organization of the human motor system is less distinct than previously thought. The perception of the M1 being a low-level area is a problematic model due to the recent research. The model of the M1 area being a high-level area instead may explain the efficiency in identifying shapes and trajectory, regardless of size. This brings us back to the original question of, how does the human motor system encode our incredibly diverse motor repertoire in an efficient manner? This study focused on the invariance of scale aspect in the human motor system. The objective of the study was to determine how hierarchical motor control is implemented by neural populations in different motor system areas. The researchers' hypothesis proposed that shape-selective and scale-invariant encoding is a general organizational principle of the neural populations of M1 and AIPS, which enables efficient encoding of the vast human motor repertoire.
Using an ROI technique, the researchers focused on 8 specific brain regions. These were the left primary motor cortex, left anterior intraparietal sulcus, left dorsal premotor cortex, left ventral premotor cortex, supplementary motor area, left putamen, right cerebellum, and left insular cortex. The main regions of interest for the researchers were the left primary motor cortex and left anterior intraparietal sulcus, or M1 and AIPS. The researchers include the other regions due to their involvement in the planning and execution of movements, allowing for differences in activation to be compared across trials. 2 control regions were also used for the comparison. These were the left orbital frontal cortex and an out-of-brain area. So, how did the researchers go about performing this study? Well, the participant base consisted of 4 males and 7 females for a total of 11 participants. The mean age of the group was 28 with a standard deviation of 3. All participants were right-handed. For the task, participants were required to lie supine in the scanner and view a back-projected screen through an angled mirror, thus preventing any visual feedback from their hand movement. Participants were required to write 3 different English lowercase letters in 2 different sizes at a 2-to-1 ratio. This was done on an MRI-compatible tablet that was placed over their waist. The letters were A, S, and N. For each trial, an audio instruction indicated the letter and scale to be executed. A fixation cross on the screen would turn green to indicate that the participant had 4 seconds to write the letter. The cross would then turn red for 4 seconds, indicating the participant should turn their hand to the starting position. Finally, the cross turned black for a randomized inter-trial interval of 4 to 8 seconds, indicating that the participant should hold their hand still at the starting position. Every fMRI session included 6 scans, each containing 7 trials of each letter at each scale. Participants were trained on the task beforehand. Bold contrast was obtained with a T2 sensitive echoplanar imaging pulse sequence, and high-resolution anatomical volumes were required with a T1 weighted 3D magnetization prepared rapid gradient echo pulse sequence. Statistical parameter mapping analysis was used to identify brain areas with significantly larger response amplitudes during letter writing than rest. The expected fMRI responses were modeled with a canonical hemodynamic response function, and a linear regression analysis was used to estimate response amplitudes for each voxel in each of the movement conditions. To analyze the fMRI data, classifiers were trained to identify letters via voxel activation. In terms of classification of letter identity, the researchers found that by averaging the activation, classifiers were unable to distinguish the letter being executed. However, in a voxel-by-voxel analysis, classifiers were able to identify the letter via the pattern of activation. In a pattern correlation analysis, the researchers demonstrated that scale did not influence letter identification. The classifiers were able to identify the letter being executed regardless of scale and at a highly significant value. Classifiers trained to identify small letters were able to identify the same letter written at the larger scale and vice versa. As well, A searchlight analysis was performed to identify other possible letter encoding regions. This technique confirmed that the left M1 and left AIPS were the two regions with the highest decoding accuracies across all classification analyses. The results show that letter identification is possible from the letter's associated fMRI response patterns, and regardless of its written size, thus providing support for the researchers' hypothesis.
Moving on to the discussion section, it is important to examine why the results of this study are relevant and how the findings are novel or different from studies done in the past. The results found in this particular study are important because they extend beyond other movement encoding findings from previous studies. Specifically, the existence of similar scale invariants in M1 and AIPS responses challenges the common description of a motor system hierarchy, where M1 is known as a low-level area that encodes only instantaneous dynamics and kinematics. That is to say, these findings are important because they are demonstrating the high-level role that M1 neural populations play within the motor system hierarchy. Additionally, the study results present a considerable advance in revealing how writing movements are encoded by neural populations in the AIPS. Although this study was extremely innovative in the way it extended beyond the findings of other studies, there were some potential pitfalls. Firstly, with regards to the methodology, classification was used. Classification suffers from a worrisome potential pitfall, overfitting. In situations where there are more voxels than exemplars, the classification algorithm may arbitrarily separate the training exemplars into meaningless groups by overfitting the large number of features. In such a situation, the classification algorithm will generalize poorly to independent data. However, to ensure that the results were not prone to classification overfitting, the authors validated classification performance by either a leave-one-out validation procedure or by validating the classification with entirely independent data. Additionally, there are always limitations when using fMRI as a measurement procedure. Although fMRI responses are strongly correlated with local neural activity, there are situations where this relationship may be less consistent. Studying encoding principles of motor neural populations by fMRI is therefore subject to limitations of neuroimaging technique. Lastly, only 11 participants were used for this study. Such a limited sample size can lead to problems involving power, effect size, and significance interpretation. So you may be wondering, what's next in this field of research? In this study, it was assumed that the letters used were faithful representatives of movements and that scale invariants would be evident in M1 and AIPS responses during the execution of other well-learned movements. Nevertheless, further studies are necessary to test this assumption. Additionally, since the authors found a new model of motor control different from the common hierarchical textbook approach, perhaps there are more models out there to explore that would allow us to have a greater understanding of areas M1 and AIPS and their role in motor functioning.